I'll show you like this. If a wood appears like this in a cross section, a circular one, in the center is the pith. Okay, so from the pith, the conduction of wood material also should happen towards the periphery, from the pith to the periphery. That's called as lateral conduction. So the ray parenchyma cells are arranged in such a manner that they run from the pith to the periphery. So they are involved in lateral conduction. Axial parenchyma is exactly perpendicular to that. So you will be able to observe the axial parenchyma more in the case of an LS. They are longitudinally placed. So axial parenchyma is storing the material in a longitudinal manner. That's how you can differentiate between axial and ray parenchyma. Okay. The next component after ray parenchyma, it is resin canals. In the case of softwoods, there are resin and intercellular canals which are present in large numbers as compared to hardwoods. This is obviously not a constant feature across every softwood, but in majority of them you find it, and it is for the protection of the wood. So they are present in the form of resin ducts. And then there are again not very constant features, but they are present in most of them. They are in the form of cell inclusions. So there can be crystals, there can be mineral inclusions, all types of cell inclusions as a result of the secondary metabolism that takes place in the wood. Some of them they may not even have it. It all depends upon the place where the plant is growing and the genetic feature of the plant. So these are the different components. Some of them I'll be highlighting and we'll be focusing on them. So before we move a little further, all of you remember yesterday, like in the sectioning part, I had mentioned that when it comes to wood, you have to take sections in three different angles. One of them is the TS, then you have TLS, followed by RLS. So first, we will take a look at a softwood across all three angles, one by one. And we will try to identify the different features one by one, how it appears in three different sections. So this one, as you can see, is a cross section. That means a TS. So in a TS, the softwood appears somewhat like this. And here I have not mentioned the name of which genus or which plant is it. It is a softwood. Slight variations will be there between different species and the features might vary. So just a general feature in order to understand the various components first. So in a cross section of a softwood, the one thing which is very prominently seen, which we discussed yesterday, that is about the growth rings. I guess everybody is able to see that. Besides that, you can observe the axial parenchyma. Axial parenchyma, you can see in the cross section, which is perpendicular to the growth rings, as I mentioned. Now, how do you identify the axial parenchyma? You can see the cells, because it's a parenchyma, they are thin-walled cells. So they are present in large numbers around. I just randomly pointed out one of them. So these are the ones which are all in large numbers having thin-walled. But there are some cells in between, as you can see, which are thick-walled. They are not axial parenchyma. Okay, let us see the other component. These are the growth rings that can be easily made out, right? Axial parenchyma, the growth rings, and they are the tracheids. That means along with the axial parenchyma itself, you will be able to notice the tracheids too. I'll show you somewhat like this. Axial parenchyma, you will be finding the cells with a thin wall like this. Tracheids, you will find almost polygonal in shape somewhat, but with thick walls. That's how you're going to distinguish. Now, in a given wood sample, they are all present together, and both of them are arranged longitudinally. That's why in a cross section, you'll be only finding their lumen. So that's how you're going to distinguish. So the ones which are comparatively thick walled, they are the tracheids. And the thin walled ones are the axial parenchyma. That's how you can differentiate. Okay. Next. Now that's the ray parenchyma. You can notice the inclination and the angle. Axial parenchyma, the lumen is visible. But in a ray parenchyma, the other surface is visible and they are perpendicular to this. You can see them in the form of lines because they are involved in lateral conduction. We also call it as medullary rays produced as a result of secondary growth. So that's the ray parenchyma. Okay, so towards the growth ring part, you will see the tracheids. They are all interspersed together. Okay, so sometimes in some species, in a cross section, in order to distinguish between axial ray parenchyma tracheids, it is difficult because in some cases the tracheids are even thin-walled. That's the reason we don't depend upon only cross section. We have to take a look at RLS and TLS also. Then the difference is very clear. Okay, so that's the ray parenchyma. Now this is the other angle. We are looking at the RLS, that is the radial longitudinal section. As I mentioned yesterday, if there's a wood cylinder, you have to take a section through the diameter. That section, when you take and observe, that's the RLS you are looking at. This is how it appears. So here, in this RLS, now see the ray parenchyma. One thing we can remember, whether it is softwood or hardwood, RLS is the one where the ray parenchyma is very clearly visible along with its composition. So in an RLS, if you are looking at cells which are horizontally present, they are the ray parenchyma. The tracheids, the fibers, the axial parenchyma, they are all present perpendicular to that. So if you want to observe the ray parenchyma very clearly, RLS is one section. Even in TLS, you'll be able to observe. Okay, so there is something called cross field pitting. Right now, I'm not touching upon that. There is another slide separately. We'll discuss that. Then these are the tracheids, as I said. They are all present perpendicular to the ray parenchyma because we are looking at the RLS, the longitudinal. So tracheids also have to be longitudinally present because they are conducting the water upwards. So they are the tracheids. Now, one more thing you can observe in the tracheids. If you observe carefully, these are the elongated ones. On the wall of the tracheids, there are these pits which are present. These are called tracheid pits. There are many tracheids placed next to each other, as you can see. They are having pits on it. This tracheid pits is also a very important feature. Right? Let me also remind all of you one more point that, as I said yesterday, the anatomical feature of the wood that we are studying can become a very important base for authentic species identification. So every such feature that we are seeing, the ray seriation, the tracheid pits, all these points are a part of the features of the wood which are unique to a given species. That's the reason I've highlighted this point as well, that is about the tracheid pits. Let's go to the TLS, the tangential longitudinal section. Now in a TLS, like one look under the microscope and you will understand any random section that this is a TLS. So what is the thing that you're going to observe if you're looking at a TLS? You will be able to see these features somewhat like this. Very elongated ones. The plain ones are the tracheids, and these cells which you can see in between, they are the ray parenchyma. So now you can recollect and compare both. How the ray parenchyma appears in an RLS and how a ray parenchyma appears in a TLS. So these are the ray parenchyma. So here we are mentioned as uniseriate. I'll come to ray seriations after a few slides, but just remember these are the ray parenchyma cells. And you can see the tracheids also. Again here, because we are talking about a TLS, it's an LS. So again the tracheids will be available to be seen in a longitudinal manner because the conduction is going on. Only in a cross section you will be directly seeing the lumen. I hope everybody must have understood how in a 3D manner you can visualize the wood now. So these three sections which I showed you was for a softwood: TS, TLS, RLS. Across all the three angles, it is for sure that you will be able to observe, measure, and analyze every feature of the wood element. That's why it is necessary to consider all three sections. Of course, along with that, even maceration. So here, as in the section what you are seeing, the tracheids and all are so clumped together. So a single tracheid you are not able to visualize completely. For that, you need the maceration technique. I'll show you photographs of the macerated slide also. Okay, So now let's go to the IVA features of the softwood. Okay, based on these three sections and based on the features, what are the different points to be seen? And there are certain physical characters also which are mentioned in the IVA. When you say the IVA features of any given wood sample, it includes even the physical features or the uh, morphological features along with the microstructure. So some all of them I have considered together and I'm showing you. The first thing, it is about the heartwood color. Yesterday we have seen the points of differences between the heartwood and the sapwood. So in the case of a softwood, now we are talking about gymnosperms. That is a softwood which grow in those conditions where the conditions of the seasons are quite extreme. And that's the reason majority of the gymnosperms, they have quite a lot of distinction in their hardwood and softwood. Majority, we are not generalizing it across every gymnosperm. So here, as it is written, the hardwood in the case of conifers or gymnosperm is either brown, red, yellow, white, or some similar shade, or it can be a combination of these colors. Yes, so one thing also we can understand logically that any one or two of these features cannot be a reliable feature for directly coming to the species level. We have to take into account all of them. It's like a taxonomic key, nothing else. So here you can say this becomes one feature. Because if I come across any random wood sample, the first thing which I will look is at the block of the wood or the log of the wood. So I will take a look at these features first before I take the section. And after taking the section, I will be looking at the internal structure as well. So these points also has to be considered. So at least we can broadly decide, okay, this could be so and so genus, right? So for example, it is yellowish brown heartwood as in the case of Pinus species, or it can be a chocolate brown heartwood as in the case of Taxus. So there is a lot of difference. So suppose if I come across a species like this, there is a high probability that it cannot be Pinus. High probability, I'm not generalizing it. A broad idea in the mind that, okay, it could be so and so. So then we can go ahead like that, right? It's similar to what we do in the case of volumetric analysis or powder analysis in the case of chemistry. You look at the chemical powder, you see the color, you take the smell, okay, you see the hygroscopicity. These are the preliminary features which you study first, and then you do the chemical analysis. Similar way over here. So that's one feature. Next, distinction between the heartwood and the sapwood. Heartwood, as we know, is in the centermost region of the trunk. Sapwood is the one surrounding it. So in some cases, the heartwood is very distinct from the sapwood. That means there is a very fine demarcation. Example, it is dark reddish brown in the case of Sequoia sempervirens, the heartwood, while the sapwood is light in color. But in the case of species like Abies, the heartwood color is very similar to the sapwood color. This is in the case of Abies. It is very difficult to make out the actual demarcation between the heartwood and sapwood. But see that in the case of Sequoia, it's very distinct. So this can be another feature. So all such observable features are also taken into account, right? So when any given sample of a wood is available, it is the duty of the person analyzing it to keep a record of each and everything. And sometimes it might be possible. See, it depends upon the age of the plant or any other feature. If the wood is affected by a fungus, suppose infected by it, then maybe there is a difference in the heartwood and sapwood. So we cannot rely upon it completely. Unless and until we do the entire microstructure and maceration, we cannot arrive at a species confusion. These are all the preliminary features, but can be taken into account. Next, we also can take the characteristic odor, because there are deposits present in the wood parenchyma. And in the heartwood, there are deposits present in the tracheids and the fibers as well. So because of those chemicals, sometimes some wood species have a characteristic odor. And sometimes what happens is, apparently the wood species might look very similar. So odor can be a distinguishing feature between them. Like for example, in the case of this one, Pseudotsuga. This one differs from larynx by its unpleasant odor, though they are microscopically similar. This is larynx, this is Pseudotsuga. Though we can also observe the difference in the sapwood color, but otherwise there is a lot of similarity between both the species. So you can distinguish between them by their odor. So the Douglas fir, that is Pseudotsuga, has got an unpleasant odor which the larynx species does not have. So one example as such. So and every other species also we can take into consideration. Everything is available on record. The literature can be understood. So this is one feature also which is considered. After the odor, now we go to the section, the internal part. So we have seen three preliminary features. Now we'll see the microstructure. So as I said, the microstructure can be quantitative as well as qualitative feature. So here the first thing is the growth ring boundaries. And yesterday when we discussed about growth rings, I said that the growth rings would be very prominent in such regions or for the plants which have evolved in such regions where the seasons are quite distinct. That's why in the temperate regions, the growth rings are very distinct. While in the subtropical or higher altitude regions, they're more or less distinct. There is a difference which you can observe. So as you can see this, in the case of Podocarpus, you can see the growth rings, but not so distinct as in the case of Pinus. So there is a difference. So growth ring boundaries also give an idea about which genus it could be. That's one other feature. Going on to the next one. Yes, in one of the sections I highlighted the trachid pits, if you observe, in the RLS. When the section was taken, when the trachedes were observed, you have observed that there are pits present on it, small openings or gaps, like small windows. So let me give an idea why those pits are present on the wall of the trachedes. Trachedes are present longitudinally, and the sap is being conducted. Now, if you are talking about a very tall gymnosperm, any softwood, there cannot be one trachea all the way from the root all the way to the topmost leaf. There will be a series of tracheads present one after the other, and the conduction is going on. It might be possible that there are certain obstacles to the flow of the water because it is going against gravity. It's like if you are sucking out any chemical using a pipette. Sometimes there are air bubbles. Everybody experiences that at least once. So you blow out entirely, remove the air bubble, and then take it again. In the case of a burette, in a volumetric analysis, sometimes if there is an air bubble, you need to rinse out the entire chemical and take in again. But what can plants do? In this kind of obstacles, the plant has to ensure that the conduction doesn't stop because the leaves require water continuously. In order to avoid that, what happens is the pits are the ones that help. So if this is one trachea like this, there's another adjacent trachea like this, arranged like this. Okay, there might be another one like this. They are all clumped together as you saw in the section. So when there are pits present on the contact walls, so even if there is a drop in the conduction, the pits will be able to ensure that the conduction doesn't stop. Water will go like this and it will go to the next one. So wherever the tracheads will end, from the pits, the conduction will go to the next one. So the pits are very, very important. And that's why the structure of the pit becomes a very important characteristic feature. Okay, so here the tracheid pits arrangement is one important feature. Now, in the case of softwoods, if you observe the tracheads properly, there are uniseriate pits also present. That means the pits are present in just one row. But there, the arrangement of the pit is not at all a point because it's just one row of pits. But if there are two or three or more than three seriate pittings, then the arrangement can be either alternate or it can be opposite. See this. This is called opposite arrangement of the pits. These are tracheads. There are pits on the walls, When you say opposite, that means they are all present in one single row from bottom to the top. That's called opposite arrangement. Similarly, if you see the alternate, now you can see the difference. In alternate arrangement, there's one here like a crisscross, one here, then on one side, then again here, then here, then here. It's not opposite. So the trachea pit arrangement is again a genetic feature. So that has to be observed in the section or in the macerated slide, and the record has to be maintained. So that's very important. Similarly, if you see the quantitative aspect of this, one can also measure the diameter of these pins. That can also become another feature. So all these points are taken into account. Examples are given to you. Right? Next. Now, in the case of tracheids which are present in the heartwood of a softwood, as you know very well that heartwood is the one where the deposits are present and they are not involved in conduction. So there can be organic deposits present. And in the case of heartwood, the deposits could be resin plugs, resin plates, or any other organic compound which might be present in the heartwood in order to plug the lumen so that the conduction does not take place. So it is reported in majorly in some of the species of Agathis and Araucaria. It appears like this. And it's again not necessary that every heartwood, every tracheid will have these deposits. It depends upon one species to another. It depends upon one individual to another where it is present. But most commonly, it is found in the heartwood region of the tracheids of most of the genera. There could be some genera where the deposits are almost nil. Okay, so that is another feature to be observed, right? This is one example of a quantitative feature now. Like if you carry out the maceration of a softwood and you separate out the different wood elements, so you can measure the length of a tracheid under the microscope. First of all, when you prepare the macerated slide, you observe under the microscope under low power. You select one tracheid nicely. It requires lot of strain to the eye, so the quality of the microscope has to be good, and the patience has to be there in order to scan the slide properly and come out, or what you can say, look at a particular tracheid very nicely. Because as I said yesterday, while preparing the macerated slide, the teasing has to be done very nicely. Only then you're separating the elements properly. Otherwise, they'll be all appearing clumped. There would be a tracheid on which there will be a fiber, or there can be some parenchyma cells scattered around. So the slide does not look very clean. You cannot create a 100% ideal slide, but majority, the elements which are important must be seen very, very clearly. So if you are able to shortlist a particular tracheid, you can measure the length under the microscope. The length could be measured using that oculometer which is available, and the respective calibration can be multiplied and you arrive at the answer which is in micrometer. So in a particular species, the average tracheid length will fall in a certain range only, based on which we call it as a short, medium, or a long tracheid. So example, in the case of AB, Spicia, etc. The tracheids which are found are either short or medium, but there are not long tracheids. So that can also be observed. So here obviously we don't take just one or two readings. When you prepare the macerated slide, there has to be a lot of tracheids measured. Approximately, it's better to take around 15 to 25 readings and then take the mean of that. So one or two tracheids going within the beyond the range is not an abnormal phenomenon. But when you take the mean and you arrive at a conclusion, then according to that you can say, yes, it is a short or a medium one. So that's how you measure any quantitative feature in a wood. You take a lot of readings. Because statistically, the more readings you take, the more you are minimizing the error which can be possible. So all these points has to be taken into account. So this is the other feature, that is average tracheid length in a softwood. Okay. Then very interesting, another feature which you can find in a softwood is something called helical thickenings. So on the longitudinal in a longitudinal section, you will find there are tracheids and sometimes there are fibers upon which there will be sclerenchymatous rings present on it. They are called helical thickenings. They provide extra mechanical strength to that particular wood. So it appears like this. Example, as in Torreya, you can see this on the tracheids. There are these ring-like structures present. They are in pairs. So how they are arranged is also another feature. Again, it is not a very constant feature, not present in every species. Okay, and it's not necessary that if you're analyzing a sample of Torreya, you will find helical thickenings on every tracheid. It might be found in some, it may not be found in some others. So, but if it is found in Torreya for sure. Similarly, in case of taxus, the helical thickenings appear different. So sometimes they are annular, sometimes they are spiral. So there are different ways in which the tracheids have helical thickenings on them. So either they are absent, or if they are present, they are in different forms like this. So we can come down to a particular genera. It could be a possibility. So helical thickening is again very important. And sometimes it might happen that if there is one tracheid, the helical thickening will be present only in one end of the tracheid and not throughout the length of the tracheid. That also has to be observed and noted down accordingly. That's why all these points together, when you note down with a very slight deviation from the actual characteristics, the wood features remain constant for a given species. Because here the only limitation is that there can be human errors while taking section, while observing, while recording the data. There could be some lack of understanding, or the section may not have been very properly mounted while making the permanent slides. The macerated slide would not have been very clean, because I've seen, like, at the college level when we do maceration, sometimes even some fungal hyphae appear in the slide. It might happen, and you mistake it to be a fiber. Even in the case of a section, it might happen. So that's why the more readings you take, the less you are, what you can say, the more you are avoiding the errors. Then you can arrive at a conclusion for a given species. Okay. Then, yes, since we are talking about softwood, there are few features which are extremely important for a softwood, and these are sure shot giving you an idea about the genera which is present. This is one of them. That's called cross-field pitting. Now, cross-field pitting, when you say, what does it mean? In an RLS, it is very clearly seen. So for a softwood, you need to take a section in an RLS and you have to observe at a specific location. As you know, the tracheids are present lengthwise and the ray parenchyma is present perpendicular to it. So wherever at the junction between the tracheids and the ray parenchyma, you will find there are these pits which are present. That is the point. That area, or the window, kind of a window which is created, is known as a cross field, and in that cross field the pits which are present are called cross field pitting. So in a softwood, if in a random sample you have to identify the wood, for all the other features you can give a rough idea that is going to be the species, but cross field pitting will give you a almost a sure conclusion which species it is. The cross-field pitting is very important feature which you will be able to see in an RLS. So now cross-field pitting are of different types. Let's see the different types of
I think there is some connectivity issue with the speaker and he has disconnected. He will join soon, I believe. Okay, so I think there is some connectivity issue with him. So meanwhile, I'm connecting so that we can reach up to him and we can find out the reason that. The speaker is connecting again. Okay, so meanwhile, our speaker is reconnecting with us. Let me tell you a few things regarding your test because many of you were asking about the pattern of your test and other details. So the thing is, you are going to have a test every week after this session, right? So on every Saturday at 2 p.m. You will be okay. So I think his connection is slow, so he is reconnecting. So I was saying that on every Saturday at 2 p.m. you will be having a test, and that test will be of MCQ type. Okay, and after that there are certain things which we need to discuss, and those things are that you will be given 30 questions from the entire 5 sessions of a week at the end of the okay, on Saturday as a test. So in that, okay, so in that exam and or in that test, you will have to secure at least 75% marks overall, combining all the 4 exams, okay, of the 4 weeks, along with 80% attendance, which is also important, right? So we will have to take care of this thing. We will have a test tomorrow, and our speaker is here again with us. Sir, are you able?
Sorry for the disturbance. There was some
No,
network
it's okay.
issue.
So, are you able to share your
Am I
screen?
audible? Yes, I
Yes,
am
of course
able
you are
right
audible.
now. So if there, yes, thanks a lot. If there is any issue, I will let you know. Thanks a lot.
Okay, so are you able to share your screen? Okay, all right, it's coming. Right, it's fine.
Yeah. Yes.
Yes.
Yes. Okay. So sorry for the disturbance, everyone. Let's continue. So I was talking about cross-field pitting. So the first type, as I said, is just 1 to 2 large simple pits, as you can see in the diagram, which are square or rectangle. Example is Pinus strobus. Similarly, in the other type, you can see this one is called as picoid pits. So as you can see in the diagram, the structure is very different. The pits have borders which are narrow, but at the same time, there are narrow slit-like apertures present on the pits. Example as is Picea and Cetrus, and that's why the name is Picoid. So we can arrive at the genus like that. Okay. Similarly, there is another one. The third type is known as taxodioid. Now, taxodioid, when you say it is, the pits are large, oval to circular, and you can see the apertures which are present inside. In the case of Picoid, it was in the form of narrow slits. But in the case of taxodioid, they are circular to oval apertures. So because it is taxodioid, example is Thuja. So here the pits are very, very characteristic of that particular genus. Then you have pinoid pits. So there are some hard pines which are present in the high altitude regions. So for that, the pits are slightly different. They are very similar to fenestriform, but in fenestriform there are 1 to 2 large simple ones. In pinoid, there are 3 or more pits which are small to fairly large, and they are often irregularly shaped. So they are hard pines. Example is the hard pine. So that's why we call it pinoid. Then you have cupressoid. Cupressoid is slightly different from the taxodioid ones, where you can see the apertures are elliptical in shape. Because of cupressoid, example is Cupressus. So there is a representative genus like that. Then you have this Orocaryoid. Orocaryoid, there are predominantly like cupressoid ones, but the pits are very, very distinctive. You can see there can be oval, there can be elliptical, there can be diamond shaped ones, and they have a polygonal outline also. The entire pit, if you observe, they have a polygonal outline. So they are Araucaria. Name itself is telling you Araucaria agathis. So cross field pitting, as I said, becomes very, very important key feature to be understood. And this gives you a very clear idea about what genus of the wood it could be present to a large extent. Okay. Then coming to the next one, this is about ray seriations. As I said, ray seriation is a feature which you can observe very clearly in the case of a TLS. In a TLS only, you will be able to see the ray seriation. So in a TLS section, in case of softwood, majority of the softwoods have only uniseriate rays, as you can see over here. Majority of the softwoods are uniseriate. Multiseriate ones are more common in the case of hardwoods. So here they are uniseriate. Very few softwoods like sequoia and cupressus, they have biseriate or even to some extent triseriate ones, not more than triseriate rays in the case of softwood. So ray seriations become another very important feature. At the same time, in the case of a hardwood, you can also find uniseriate rays, but they are less in number as compared to multiseriate rays. So ray seriation is very important feature. So here we also take the quantitative data for the ray seriation. We take 10 readings, like for example, if I am selecting one particular ray, suppose if I am selecting this one. So what kind of ray features I will note down? I will note down the seriation, which is 1. That means just one row of cells. I will count the number of ray cells from the top to bottom for that particular ray. Maybe 6 or 7 or whatever. And then at the maximum width point, I will measure the width of the ray under high power. That is also an important feature. So ray width, ray seriation, ray number of cells for every given rate is taken. So 25 such readings can be taken easily, and you take a mean of that. So these are very important features. Similarly, here if I measure this one, so I will write the ray seriation as 2, and this at this maximum broadest point I will measure the width. That's the ray width. So for any given ray you select and you take all the features of the wood accordingly. Okay. Then coming to something called intercellular canals. For the softwood, this is the last one I'm showing you. As we saw in the beginning itself, softwoods have lot of these resin ducts, intercanal canals, and this kind of ducts which are present, but they are arranged in two different ways. Okay, they are tubular arranged ones longitudinally, and they produce some secretions which are providing kind of a protection to the wood species. So either they can be present axially, as you can see. That means you will be able to see this in a cross section. It will appear very similar to vessels, but they are not vessels. So they will be observed either axially, as in the case of Picea, or they can be seen radially, as in the case of Pinus. So either they can be axial or they can be radially present, depends upon the species. Again, it is not a constant feature, as I said. So every feature has to be taken into account, whatever is observed, and then accordingly the conclusion can be drawn. So that was all about the softwood. Many more features are there, as I said, but the list is very huge. So the major ones I have shown you. Similarly, coming to the hardwood. Now, if you have understood the softwood properly, the hardwood will not be very difficult to understand. Let us take a look at the features. The most important feature which you find in a hardwood are the vessels, because the vessels are the conducting elements. They conduct sand upwards. And yesterday, as I mentioned, because of the presence of vessels in the hardwood, then you have fibers for mechanical strength and support. So more amount of fibers you will find in the case of a hardwood. Then you will find the ray parenchyma, again for lateral conduction. You will have axial parenchyma for storage, very similar to the softwood. You will have different types of cell inclusions present in various forms. So these are the different components. Okay. So taking a look at the cross section also. If you recollect the cross section of a softwood that you observed, the growth rings were very prominent, axial parenchyma was seen, and a lot of tracheids were seen. Here in the cross section of a hardwood, what are you able to see? You are able to see the different vessel pores. Many of them are seen here. You can also see the different fibers. Now, fibers can be understood by their thick walls. There we saw the thick walls for tracheids. Here the fibers will have growth ring boundary, again in a hardwood. And then there are ray parenchyma which is present. You can see the ray parenchyma more distinctly in the case of a hardwood. So in a cross section of a hardwood, these are the features you can visualize. So one look at the section and you understand, yes, it is a hardwood. It cannot be a softwood. Right? Similarly, you can also see the TLS, that is the tangential longitudinal section. Now in a TLS of a softwood, you remember you had visualized the ray parenchyma. The ray seriations were seen. In case of a hardwood, you can also see the ray parenchyma in the same way, but here you are seeing the ray seriations are more. Along with that, you are seeing the axial parenchyma, which are surrounding the vessels. You are seeing these are the vessel elements present end to end like this. In a TLS, you will be able to see the conducting pathway. Then you can see the different ray parenchyma cells followed by thick walled fibers. Tracheids are also intermingled along with the fibers, but they are very less in number in a hardwood. Okay, so one look at the section and you can make out, yes, it's a TLS. Similarly, if you look at the RLS of a hardwood, Now you can see the ray parenchyma. It is similar to that what you saw in a softwood. They appear horizontally present, and from a softwood and a hardwood you can distinguish by observing the presence of these vessel elements. So in an RLS you can see the vessels, the fibers which are present along with that, and the ray parenchyma. So in an RLS, the ray parenchyma becomes very important features. So we have been talking about all these different wood elements. So let us take a look at how these wood elements appear in a macerated slide. Okay, this one shows you the different fibers. See how they're nicely separated out. You can see the fibers from end to end. Some of them fibers can be understood by that nice tapering ends. This is taken under low power. If you observe it under high power, you can see the thick walls of the fibers also. So you can keep the oculometer accordingly, and you can measure the entire length of the fiber from one end to the other. And you can note down the readings. So you have enough number of fibers on the slide. You can take the fiber length, 25 such readings, and take a mean. You can also take the fiber diameter under high power and take 25 readings. So macerated slide gives you a very clear picture. This kind of entire visualization of the fiber is not possible in any section because they are all clumped together, intermingled together. See this. This is a trachea. Now this is cannot be called as a very good slide, but yes, you can visualize the entire trachea as it is from end to end. They are thicker, they are broader than a fiber, and there are pits present on the wall of the trachea as you can see. That's how you understand it. And you can differentiate it with a vessel as well. A vessel has got open pores of openings on both the ends. Tracheas have tapering ends. That's how you can distinguish. There are pits on the walls also. So this is just to show you how these features appear under the macerated slide. Now in this vessel, as you can see, these end walls. This is not possible to see under a section. That's why you need to do the maceration. Okay. Then let's see some of the important features. Okay. Now in the case of a hardwood, the growth ring boundaries can be demarcated by three different ways. Okay. See the first one. In case of a hardwood, either fibers or tracheas, okay, they can be present which demarcates the growth ring boundaries. Like you cannot distinguish based on the vessels. So as you can see, if this is a growth ring boundary, you can see here the fibers and the tracheas are thick walled, while here they are thin walled. So that means the demarcation is because of thickness or thinness of the fibers and tracheas. Example in the case of Laurus. This is one way you can demarcate a growth ring. Second way is by the vessel itself. Okay, you can see the difference in the growth ring boundary. Some vessels are very broad, big ones, while some others are thin ones, or small, narrow ones. So this is very clearly seen in the case of teak wood, that is Tectona grandis. Third one is because of the parenchyma. Okay, so parenchyma can be present as a demarcating feature between growth ring boundaries. So either it is because of fibers or tracheas, or it is because of vessel diameter, or it is because of the parenchyma which are present. So the feature can be observed accordingly. Then the next feature in the case of a hardwood, based if the growth ring boundaries are decided based on the vessels, then wood porosity can be distinguished for them. Okay, we are talking about growth ring boundaries decided by the vessels. Okay, so it can be classified into three types. Wood can be ring porous or semi ring porous or diffuse porous. You can see the difference for yourself. In case of ring porous one, you can see the vessel diameter has got one broad range over here, which is suddenly becoming small vessels on the other side. So the vessels which are big in size are forming a part of the early wood. while the ones which are small, they are a part of the late wood. When the distinction between the vessel diameter is so apparent, we call it ring porous. On the other hand, if you observe the diffuse porous, you can see that the vessel diameter throughout the growth ring is more or less constant within the same range. So we call it diffuse porous. And if it is in between both of them, you can see there are slightly larger vessels, but the late wood vessels are not very small. But you can make out the difference in the range. Then it is called semi-ring porous. So that's how you can understand difference between ring porous, diffuse porous, and semi-ring porous. Okay, wood porosity is possible. That's the reason only for hardwood. Okay, coming to the next one. Now, if you take a cross section of a hardwood in a TS, observe the vessels, how they are arranged. See this. They can be arranged in the form of tangential bands. That means you can see they are present horizontally in lines, like in bands. They are called tangential bands. On the other hand, in some other species, you can find they are present either diagonal or radial. That means exactly perpendicular, or you can say parallel to the rays in long lines. This is called diagonal or radial. Some of them are cross ones, so diagonal, and they can be radial, as in eucalyptus. Okay, or they can also be present in the form of a dendritic pattern. Dendritic pattern means like there will be clumps of vessels at one place forming a branching structure, and there will be some portion of the cross section where there are no vessels at all. This can also be a possibility. So arrangement of the vessel pores as seen in a cross section can become very important features. Not only that, see this, groupings of the vessels, again which you see in a cross section. So how do you observe this? See, if majority of the vessels in a cross section are individually present so that their boundaries are not touching any other vessel, more than 90% of the vessels, if they are like that, we call it exclusively solitary. Solitary means alone, as in ficus. Similarly, they can be radial multiples. From the picture, I think it is easy to make out. They can be in the form of radial multiples of 4 or more vessels together. Example, strychnos. Or they can also be in the form of simply clusters. That means you cannot call it tangential or radial or something. It is just like one small bunch at one place. They are called clusters. Can also become a very important characteristic feature. So because of environment or because of something, these features will not change. They are qualitative features. For a given species, these will remain the same. Right? Okay. Now, if you see a vessel, now I have shown you individual vessel in a macerated slide, how it appears. So vessel is a cylindrical structure, but towards the end, there are those openings. Those openings which are present, or the pores which are present, they are called perforation plates. So from one vessel to the other, adjacently when they are connected, the perforation plates are the ones which are forming a common point of contact. And they are of different types in vessels. The simplest one is called simple perforation plate, where the opening is just one hollow opening, nothing else in it, as in the case of Pterocarpus. Similarly, sometimes the perforation plate can be scalariform. That means there are ladder-like structures, as you can see, instead of just one open. This is called scalariform. In the case of shrubs and all, you will find such species. Okay, Rhizophora, you can see that. Then this type is known as reticulate. The word reticulate is very commonly known. It's like a networking pattern. So here you are able to see a network-like pattern in the perforation plate. And then you can see this is called foraminate perforation plate. Foraminate means simply like small sieve pores. You'll be able to see it like that. So perforation plates are of 4 different types. Now in any given hardwood species, It is not necessary that it will have only one type of a perforation plate. It might be possible that larger vessels will have simple perforation plate, smaller vessels may have scalariform ones. So every species has got its unique combination. But the type of the perforation plate for a given species will not change due to environmental reasons. That's why it is a very important diagnostic feature. Okay. Similarly, if you observe the walls of the vessels, okay, in a macerated slide or even in a section, on the vessel walls, there are pits present. In the case of softwood, we saw the trachea pits. Same way, in the case of vessels, we are looking at intervessel pits. So intervessel pits, again, they are very small in size, which you have to observe under the high power and again take their diameter for the range. So the size of the IVP is a diagnostic feature. For a given species, the size range will remain fixed. Of course, you have to take 25 readings and all and then come to a mean value. Okay, so the arrangement, see this on the walls of the vessels. You can see very nicely they are present in an alternate pattern. The functioning of the intervessel pit, the conduction remains constant. Okay, here we call it intervessel pits. So alternately arranged, or it can be arranged in an opposite manner. See very clearly. Next to each other. The shape of the IVP is a diagnostic feature. Okay, or it can be in the form of scalariform pattern again. Now one should not confuse between scalariform perforation plate and a scalariform IVP. IVP are the pits present on the walls. Perforation plate is towards the ends of the vessel. So the more you read and understand, it will be clear. But these pictures are showing you how different they can appear. Right? Now, when it comes to IVPs, in some species or maybe in some families, there is one very interesting feature that is observed in the IVPs. We call it as vestured pits. IVPs show a phenomenon which is known as vesturing. What do you mean by that? It might be possible that this is kind of an adaptation in the plants, we can say, that it might be possible in some plant species, there are some highly infectious, or we can say any contaminating production pathway. So the plant wants the conduction pathway to temporarily stop in that region. So these same IVPs will invaginate and the walls will close. So this is taken under a high power and this is the electron microscopy image of the same. You can see how the IVPs have almost closed down, blocked. So there is a projection from the secondary cell wall of the IVPs. Such features are called vestured pits. They are more commonly seen in some families like you have Combretaceae, Myrtaceae, Rubiaceae, etc. And it's not only on the intervessel. It can be on other pits in the plants as well, vessel ray pits, or the pits between vessel and axial parenchyma or interfiber everywhere. But more commonly seen in the intervessel pits. Such pits are called vestured pits, and the phenomenon is known as vesturing. Again, very important key feature. So this is an SEM which I had taken from my samples. It's on Tamarindus indica. Okay, so scanning electron microscopy shows you really beautiful structures. I had done the scanning electron microscopy for my samples from the Punjab University at Chandigarh. I carried some of the samples for the SEM to be done and showed really that was a wonderful experience. When the samples were given, we were in the lab and the in charge over there who was handling the instrument, that person, when the instrument started, if you have done the SEM, you might be aware, it's like as if you are entering into the wood yourself. It's taking you in a ride. Okay, this is the vessel. You will go from here, you will come out of a perforation plate, you will go to another vessel, you will come out of an intervessel pit, go to the adjacent door. Very interesting one. So at some particular places, if you observe some unique phenomenon, you click the photographs over there. This is one of them. Okay. Then when it comes to vessels in the wood, you take the macerated slide and you quantitatively measure a lot of features. So a lot of features like you have the entire length of the vessel you can measure. Okay, you can measure the diameter of vessel, like I take randomly 25 reading per kilometer and measure the diameter. Similarly, I can also measure the vessel wall thickness, or I can measure, as I said, IVPs. The diameter of the IVPs can be taken into account. So a lot of such quantitative vessel characteristics can form a part of the study. For a given species, the values fall in a certain range, which is very important. Now, when it comes to vessels, this is another very interesting feature. It's called tylosus. As you know very well, in the heartwood, the conduction does not take place. The conduction generally takes place in a sapwood portion. So when the wood is in a very mature stage, and if it is a heartwood, the conduction has to stop. But there are vessels present over there. So those vessels, the pores have to be filled up. So either there are organic deposits which are plugging the vessel lumens, or it can be because of the invagination of the walls of axial parenchyma or the vessel walls into the lumen. So it gives rise to structures like this. This phenomenon or these structures are called tylosis. So the tylosis are characteristic feature very clearly seen in some species, and sometimes the tylosis can be very thick, highly lignified ones. They are known as sclerotic tylosis. So in the under a magnified lens also, if you observe, you might be able to see tylosis like this. And under a microscope, you'll be able to see tylosis this way. This is from a section I have taken from Shoria robusta. It is a xylarium specimen I had taken and carried out the sectioning. This is what I could observe. Okay, so tylosis function is what? In order to block the lumen and the heartwood region so that the conduction from there does not take place. Okay, and in fact, it's like if the lumen remains open, it can also be prone to infections, and also that's why it has to be plugged up. That's called tylosis. Okay, yeah. Now coming to axial parenchyma, which is commonly found in the case of softwood also, and in the case of hardwood also. So here, when you say axial parenchyma, first of all, what it is? It is the parenchyma, normal parenchyma cells, which you can observe in a cross section present surrounding the vessels. Axial parenchyma are the cells, parenchyma cells, which you will find surrounding the vessels. So here in this cross section, as you can see, the parenchyma, the small parenchyma cells which are surrounding the vessels. That's called axial parenchyma. Now there are two types. First one is apotracheal, second one is paratracheal. One difference which is visibly seen is that apotracheal parenchyma surrounds the vessels, but they are not exactly touching the vessel elements. In the case of paratracheal one, you can see they are touching the vessel elements in a cross section. That's the difference. There are further types under this, like the details are not read as of now. You can always read. So apotracheal example is Santalum album or sandalwood, and paratracheal example is Delanium. The further types have their own specific examples, but in some cases, the axial parenchyma looks really beautiful. As you can see, these are eye-shaped structures. That's called alliform parenchyma. Sometimes they are joined to each other, forming alliform confluent parenchyma. So they look very nicely under the microscope, and it's a qualitative feature. And some cases, the axial parenchyma under a cross section can also be seen as bands. We call it banded parenchyma. So the bands can be like complete bands of parenchyma. Sometimes they can be 2 or 3 cells thick. Sometimes they can be many cells thick. See this. As in the case of Ficus retusa. Big bands. These are all entire bands of parenchyma, completely covering the vessels. So they can be more than 3 cells wide, or in some cases, as you can see, the bands can be narrow. So all these features are also taken into account. How the axial parenchyma is arranged. Okay. Then similarly, yeah, the ray seriations as we saw in the case of softwood also. Okay. Now here in the case of hardwood, the ray seriations and width have lot of variations. See the first one. In some species of hardwoods, you will find exclusively uniseriate rays, similar to softwood. But here you have to understand it by the presence of vessels. Okay. Or it can be small thin rays with 1 to 3 cells. Okay. Like that we can keep on increasing in number. It can be 4 to 10 seriate also, as you can see. Not all, majority of them. Again, you need to take 25 readings around and then come to a conclusion. Okay. There can be broader ones also. Larger rays commonly more than 10 seriate. Such huge rays can also be found. Or there can be a different concept where uniseriate rays will have cells which are large in size. Multiseriate rays in the same wood will have cells smaller in size, so that the overall width of both the rays remain the same, more or less same. So if it is biseriate, the ray width is the same as that of uniseriate one. Even this is possible. So such flexibility and variations you find in the case of hardwood when it comes to ray seriations and ray width. Okay. Then, yeah, this is again very important feature in the case of a hardwood, which you don't find in a softwood. In case of a softwood or even in hardwood, the ray composition can be understood only in an RLS. Okay, so this is an RLS which is taken, and as you can observe, the cells of the rays which are seen to be present horizontally are of different types. You can see there are 3 types of ray parenchyma cells in hardwoods. The first type is called square cells. Name itself tells you the shape of the cell. The cells are almost squarish in outline. Second type is upright. You can find the cells are finger shaped, like in the case of palisade cells of a leaf. And the third one, they are more of elongated rectangular ones, which is called procumbent cells. So in the case of a hardwood, you will find ray composition consisting of all 3 types or any 2 types or any 1 type. But in softwood, all of them are only of 1 type. Most commonly it is the square cells. That's how the major difference between softwood and hardwood can also be understood. So in case of hardwood, you will find rays which are homogeneous or heterogeneous, depending upon the combination of the ray parenchyma cells, which is clearly seen only in an RLS. You need to remember the 3 types: square, upright, procumbent. Okay. Then, yeah, this is another very beautiful architecture of a wood which can be seen in some species. Something called storied structure. Okay, when you say storied, it's like if you're aware of something called as an apartment system or a tall building, okay, which in big cities we see. So there is first floor, second floor, third floor, all present in a same line. Similar thing you can observe in the wood also sometimes. See this. Sometimes in the section, if you observe, all the fibers are longitudinally arranged in such a way that their ends are coming in a straight line. Majority of them. So they are called storied fibers. They appear very beautiful under the microscope. Sometimes the ray can be storied. See this. Okay, the rays are very beautifully arranged exactly uniformly in line with the vessels also. Most of them. Or sometimes you will find the vessel elements are arranged nicely in one line. You can see the simple perforation plates here. These and these. That gives you an idea that they are vessels, all present in one line. So this architecture of the wood is a genetic feature. So some species are having this. So studying such features under the wood when it is storied and all like this becomes more interesting. And collection of data from the wood becomes very easy if it is a storied one like this. Okay, so this also can be understood. And lastly, we come to something called mineral inclusions. Okay, now here in the case of a hardwood, it is a possibility that the plants will carry out certain physiological process and it will metabolize some harmful chemicals into immobile forms and store it inside the cell so that it does not cause any damage to the plant. So there can be of various different types. You can see it in the form of raphides or in the form of crystal sand, or there can be cystoliths as in ficus, or there can be silica bodies. Okay, some of them I can show you. You can see these are prismatic crystals. See under the microscope in a section how it appears. In each cell you can see one crystal present or maybe two crystals present. They appear very beautiful. And for this, you have polarized microscope which you can use, in which this appears really amazing. Polarized microscope means it's like you can create in a 2D manner, like the section part will go in a background and only the crystals will be highlighted. So some of the species have so many amount of crystals present in it that when the you observe it under the polarized microscope, it's as if like there are nice diamonds and pearls present inside the section. They appear very beautiful. And if you observe that under scanning electron microscope, then the beauty is like endless. This is one entire crystal, as you can see in Tamarindus indica. Okay, so legumes generally have a lot of prismatic crystals within it. Right? Similarly, see this drusus. Prismatic crystal, as you can see, there is one or maybe two crystal in one cell. If there is more of, you can see that in the case of Terminalia fetapa. You can see in an SCM also there are a lot of it present. Aggregation. Okay, there's one paper which is published in my name, where I am also the author. I'll show it in one of the slides now. And thirdly, styloids. That means in the form of long rods, needle-like. Okay, so in case of Terminalia, you can find that also. You can see how it appears under the microscope in SCM. Bunch of this. So this crystal studies or mineral inclusion studies can also become a separate study when it comes to wood. And a lot of information can be obtained out of it. Okay, so we have discussed about the different microscope structure features of a hardwood and a softwood, and wherever it was applicable, I have told you what are the differences which are present between them. So that I have shortlisted over here, like presence or the absence of vessels, the difference in the growth rings, the difference in the ray parenchyma, the seriations of the ray parenchyma, porous wood, non-porous wood, all the points are compiled, and there can be many more points also which you can include, right? So the microstructure features of a hardwood and softwood, on this points which I have shown you, about a dozen of them for each one I have shown. Based on that, now you can understand how different the wood appears internally in a gymnosperm and in an angiosperm, right? So let me take you some interesting photographs and all, and then we can come to an end, right? So these are some interesting micrographs which I had, which I thought I can share with all of you, right? So just some random pictures, like you don't need to know the name of the species and all. So here, this was the section. Like you can see how beautifully the vessels are present in clusters, aggregates of vessels. Okay, and the ray parenchyma seems to be highly multiseriate, right? So from a cross section, you can make it out. So for taking now this kind of photographs which I'm showing you, for that again, it's not like you take a microscope, you keep your mobile phone on top and take a picture of something. At a higher level, you cannot do that. So there are softwares available. Okay, so AxioVision is one such software which was available in my laboratory, using which I have taken these pictures. Okay, so it's like a complete package. There is a microscope having an inbuilt light facility, and which is connected. It has got a camera and it is connected to a computer. So you have to click it on. Whatever you are looking at, you have to place your slide just like any other microscope. You have to observe inside, check the brightness and all, come to a proper field where you want to click the photograph. Then with a knob, you can click on the screen in the computer. So the same thing which you are seeing under the eyepiece, you will be seeing it on a screen in the computer. And then there are options available to change the brightness, contrast, and all those settings. Even the colors also to some extent you can change. So you can try that and see whichever is suiting best, and all the features are clearly visible. Where when after which you can click on snap. That means the section will be clicked. And there is an option available to keep the scale. This is what you can see written over here. So the scale will give you an idea that the section is taken under which power, 10x, 40x, or 100x, or maybe whatever. Okay, so that has to be inserted accordingly, and then the photo has to be taken. Okay, so this kind of instruments or softwares are also available. Then this is a macerated slide showing you a very clear and entire vessel element with the end walls also, right? Then this is the SEM image. The same vessel element, as you can see, how nice perforation plate can be seen. This is an RLS if it is viewed clearly. You can see the, if you use an SCM for this wood analysis, you can actually enter into this and come up from some other place. Okay, that's a very beautiful one. See under the high power, now you can see the scale is a longer one. That means it's a high power. Okay, observe these intervessel pits or the IVPs, very beautifully arranged. The intact vessel was not available, but the IVPs appear very nice. That's why I'm showing you this one. Very beautifully, you can see the primary wall as well as the secondary wall of each IVP. Okay, and then this is the fiber. You can see the wall thickness can be very nicely seen. So while taking readings for fibers like this, what we do, we keep a high power. You take the wall diameter from end to end as one reading, and you take the diameter of only the lumen. So suppose this is A and this is B. So fiber wall thickness would be A minus B divided by 2. That's the value of your fiber wall thickness. Like that you take for 25 such readings and you come to a conclusion. Okay, so it's a tedious task, but it's very interesting. Okay, this is the paper which got published in my name, and this paper gives you a complete detail about my PhD work. As I said, like in a broader way, if I tell, my study area was in the iron ore mines of Orissa. There are very old iron ore mines present over there and surrounded by huge forest, where there are trees which are growing for a lot of decades. 100-year-old trees are present, even more than that. So I selected that area. I selected the hardwood tree species. I collected the samples. How I collected and all I have shown you yesterday, and then analyzed the wood features, all the features according to the IVA list for hardwood, and I have compared this with the xylarium specimens which are available in our xylarium and FRI, and for the same species. So if I collected Mangifera indica wood sample from the forest of Orissa, I have compared that with the Mangifera indica xylarium sample available over there, and then statistical analysis was applied in order to observe that hundreds of years of mining activity in the iron ore mines, whether it is having any impact on the growth of the wood. So I have done this work for 22 different species. Many samples, many sections were carried out, statistical analysis was applied, which has given rise to good findings. So out of the 22, I have shortlisted one species which is very tolerant and one species which is very adaptable. So I can, the implication of this research is that wherever in such mines, whether it is in Orissa or any other place, if the mining activity is completely done and the restoration of the mine has to be done by afforestation programs, we can suggest some species to be grown over there, rather than randomly selecting any species, and preferably not the exotic ones. So that becomes an implication based on these criteria. So there were 3 criteria I did. One of them was the wood microstructure. Secondly, it was the iron ore and the iron element content, which was taken up by the species. And thirdly was the specific gravity of the species, the wood samples. All 3 together were taken into account. Statistical analysis was done, and we arrived at a good result, and it got published in one of the standard journals called Trees, which is one of the top 5 journals for plant sciences. So If anybody wishes, one can go through these papers. We'll come to know what kind of work can be carried out. Right? This is one of them. Similarly, there is one more publication, and which is actually a new report. So one of my colleagues, Ms. Ankita Gupta, she was working on the coal mines of Jharkhand. Similarly, what microstructure was being carried out, and she came across one Mangifera indica species. We were working together. Mangifera indica species collected from the coal mines, and in which she saw that there were druses which are present in the wood. And based on the available literature, presence of druses in Mangifera indica was never reported earlier. So it was the first observation. So that was a very drastic change which has taken place, and that's why it has got been published over there. So we carried out the SCM and all, and we confirmed the same. So this is another publication based on that in Journal of Tropical Forest Science. It's a Malaysian journal, right? So like this, publications can also be available. Okay, and at the end, of course, I would like to acknowledge, like, because I was able to study and explore so much into the wood science. Very interesting features were seen. Got a lot of learning experience by staying in the forest, interacting with the villagers over there, how they helped me in sample collection, like how they, because as I said yesterday, the increment borer was very tedious in order to put it inside the trunk and then turning it back, doing it for a many tree species in a single day. Okay, and every tree species is like, every individual tree is not very easily accessible. Okay, sometimes you have to stand on a slope or maybe on a rock. Okay, other animal species can be there. So all these things were there. So got a very beautiful forest experience, ultimately giving really good results. So one can do such kind of work and come out with very like whatever I did was actually an innovative and a novel work. Such work was hardly carried out anywhere in India. Okay, and the microtome which I showed yesterday, that was available in this MS University Vadodara. He is the PI of that lab. She is Mrs. Ankita Gupta, my colleague. Dr. K.S. Rajput sir, he has helped us a lot tremendously in this work. And she is my guide, PhD guide, Dr. Sangeeta Gupta madam. And this is during my convocation with my proud parents and my sister. And then he is, because as I said, we'll applied a lot of statistics in my work. So understanding statistics to that level, applying it for so many species and plants, required a lot of good expertise. And I was fortunate enough to have the expertise and the guidance of this statistician, Mr. Raman Notial sir, who was a statistician at ICFRI Dehradun. And no doubt, after all this got over, since 4 years, I'm here in Bombay, teaching in this college, SIS College of Arts, Science and Commerce, autonomous, since 3 years. And I'm here working as an assistant professor, encouraging the young minds also by sharing my experiences, so that the thing which I always advise my students is that whenever it's about a classical subject like botany, okay, the exploration should be endless. The learning part should be with an open mind. That is very important. Okay, one should not keep a stereotypic tendency that this field has got scope, that field doesn't have scope, and all that narrows down your thinking. Okay, there are plants outside, so of course there are things to learn, and the nature is so vast, the plant world is so vast, that we are not yet completely aware about 100% of the story behind. You never know, when you explore, you can come out with something new which has never been reported earlier. One more example I can give you. During my study in one of the forests of Orissa, I came across a place where there was one tamarind tree and a mango tree growing together. Together in the sense, now it is Tamarindus indica and a Mangifera indica. Both were very old trees. They were growing together, means their bases were connected like this. And then the trunks were growing separate. At one point, again, they joined and they are forming independent branches. So somewhere internally, they are connected. Like this much what I saw, and I spoke to the villagers also. So what they said was, like, whenever the tamarind plant produces the fruit, it has got a slightly different taste, which is slightly towards mango, and vice versa in the case of the mango fruit also. Of the day, maybe you can, well, somebody can go collect some samples, do some physiological analysis, understand how actually whether the wood is joint or not, whatever the villagers are saying is true or not, that could be a possibility. And according to what I spoke to the villagers, both the trees were more than 100 years old. So this kind of an intercaste marriage between tree species was something I found it for the first time. You never know, it might be possible. So exploration has to be with an open mind. You see, you observe, and then learn. And as far as the career is concerned, everybody needs to carve their own path. There is no fixed path available. Go one step ahead, the next step will open up. That's how you can make a career. So that's my advice for this today's generation, that explore, create a name, do something, come out with something with flying colors. I hope it was an enjoyable experience into the world. Thank you very much, everyone, for the patient listening. And we can be discussing anything more if you want. Thanks a lot, everyone. Yes, sir. Devinder, sir. I am done with my part. Thanks a lot. Devinder, sir, I am done with my part. Can you please continue?
Thank you so much, sir.
Thank you. Thanks a lot. Thank you for the patient listening. Okay, if I can just interact with the audience. Are there any participants who are from outside India? Any students?
Bangladesh.
Okay. May I know your good name, please?
My name is Amjad Fordul Islam.
Okay, thanks a lot for attending. Hope you enjoyed it.
I'm studying in Dhaka College, affiliate in Dhaka University.
Okay, that's great.
So I need your mail number, please, if you are interested to send you mail number.
Devinder sir will be forwarding my contact details to anybody who requires it. You can be in touch with Dev sir. He will be providing all the required details.
Thank you, sir.
Anyone else from outside India? Okay, majority of the students are from Delhi University or any other part of the country?
Sir, I'm from West Bengal, Calcutta University.
Okay, that's great. Your good name?
Sir, Nisa Tandon.
Okay, thanks a lot. Hope you enjoyed the session.
Yes, sir. Thank you, sir.
Thank you.
I am from.
Thanks a lot. Your good name, please?
My name is Himadri Shekhar Patta.
Okay, thank you. Hope you enjoyed and got a good learning experience.
Yes, sir. It's a very informative session on science, and I got interesting opinion.
Okay. Okay. So good to, it was a really good one from other countries like Bangladesh. So this interaction was really fruitful and I enjoyed delivering this lecture to all of you. Just contact them, sir.
Thank you so much, Dr. Vishnu. So
Thank you, sir.
again, it was indeed a very nice and interesting lecture today as well.
Thanks a lot. I can see there are
So
students from Tamil Nadu, Rajasthan, Kerala, Punjab, West Bengal, and even from Bangladesh also.
yeah,
It
so
was
we
good
have a very
to have
interesting.
everyone under one roof. Yes.
Yes. And interestingly, because they all are from different regions, they must have seen these variations among tree
Yes.
species
Yes.
and their
Correct.
wood structures as well. I think we should start with some questions from the audience.
Yes.
So there are a few questions. Okay. Okay, so Ayush has asked something today, but I don't know what he's actually trying to say. He's saying that I had a question. There is a certain point of year where tree stem widening stop, but trees still live to face with respective seasons. Therefore, how after to calculate that tree seasonal condition. So I don't know what he's actually trying to say, but I believe he's trying to say that when there are when there is no difference in the seasons, that then I think he's quite asking about the growth rings maybe.
Okay. See, that's what, as I said yesterday also, like if there is no much distinction between the seasons, the growth rings generally do not come out very prominent. But it is not only the environment that plays a role over here, because if it is it is a condition for one or few tree species or tree individuals, it might be some kind of an adaptive feature. But at the same time, genetics also plays a role. Okay, so if the particular tree species has got, for that to change over a period of generations during evolution, will require a lot of constant exposure to the changed environmental conditions. Only then it will be inherited. So that has to be studied and in depth further. Only then one can arrive at some conclusion.
Okay, I don't think we have much questions today. Maybe because all the students they
Okay.
were very much they were very keenly listening to you.
Yeah, actually, it needs more in-depth study, and yeah, the more they will read the books and all, maybe they will come up with some more questions.
Right. Okay, so now we have one question from Kirti 259. She has asked that, sir, there are organic deposits in hardwood. So, is there any method to extract those deposits, or are there any deposits that are commercially useful?
One such example is our sandalwood. That is what we use medicinally, even for cosmetic purposes. So it is nothing but deposits in the hardwood that are obtained. So you can take it off in the form of a paste or make a decoction or a solution, and you can use it. Other such wood species can also be studied further, and probable medicinal uses can also be explored. But the most commonly used one is this sandalwood.
Okay, thank you, sir. We have another functional difference between within the three types of ray parenchyma cells.
Okay. So functionally, they are not much different as such, because the dimensions of the ray parenchyma cells are different in these three types. So the only difference we can say is the amount of the storage that can be carried out. Okay. So otherwise, it has got no functional difference. It's a genetic feature, and which is leading to some diversity. So homogeneous, heterogeneous, there are many further types. So each species has got its own combination. So it's nothing but the diversity and the genetic feature which you observe. It's like in case of a leaf, whether you have a crenate margin or a serrate margin, functionally there's not much of a difference. It's the presence or the absence of the chlorophyll in that region. Same way for the wood ray parenchyma.
Right.
Good
Thank
question.
you. So we have another question from Gurjan 211. He is asking, sir, how does tylosis help plant in disease combating?
Yeah, because see, tylosis means the vessel pores are plugged with invaginations or some deposits within it. So if at all any fungal hyphae, majority of the diseases are by fungi. So if the fungal hyphae try to penetrate the wood, So because of these stylosis and all, they will be prevented from going further and causing more damage. So that's how the disease prevention can take place.
Okay.
Yes.
So thank you. We have another question from Shaheen 442. She has asked, Pterocarpus anglolensis is also known as bloodwood,
Correct.
and it releases red pigment when wounded or cut. So
Yes.
what's the reason for this?
That is because of the presence of that red colored sap. It has got some physiological process by which that particular compound is produced, and it is a part of that parenchyma as well as the vessel. So whenever it is given a cut, it oozes out. And sometimes what happens is in some wood species, even in hardwood, now Pterocarpus is a hardwood one. So even in the case of a hardwood, there are some kind of canals which are present, intracellular canals or maybe latticeous or sheath cells or tile cells, perforated ray parenchyma, all such possible canals and ducts can also be present. So this kind of products are found and they are stored and transported within these canals. So it is a unique one where you get this red colored compound. It is a secretion which is produced by this. There are many
Right.
other examples like that. There's one Terminalia species also which gives out water, which oozes out when it is given a cut.
Okay.
Yeah.
All right. So we have another question from um, Murugeswari 281. Uh, they have asked, sir, what is cross fiber pitting?
It is actually cross fiber pitting in the sense like when fibers are placed adjacent to each other. So wherever the walls are overlapping, there are pits present. So these pits which are present on the walls, which forms a connection.
All right.
Yeah.
Bhupen has asked one question that, sir, is there any plant that possesses medicinal values of its hardwood? The question arises, as you said, these are formed of deposits.
Correct.
So I think there are
Sandalwood,
many
yeah,
examples.
sandalwood as I gave the example.
Yeah.
Yes.
Now we have another question from Kanika 252, that how to combine pre-reading data with incremented.
Okay. Now see, use of dendrometers and all requires some expertise, which practically I have not done any time. But it's like you need to keep on measuring the formation of the data is nothing but the measuring the growth rings, how it is formed. Okay, now when you know that it is concentric, sometimes at some places the width of the growth ring, the same growth ring can be different on one side, can be slightly different on the other side. Okay, so that can be understood properly when you take the data using dendrometer. I can recollect one example over here. Like as I said, my colleague Mrs. Ankita, she was working on coal mines of Jharkhand. So when we were over there, in the coal mines of Dhanbad, which are one of the largest coal mines in the world, it's a coal capital. So there are a lot of coal explosions which keep taking place. The blasting of the mines have to be taken place. So there were some eucalyptus species over there which were planted, and it was exposed to those explosions when we were explosion after every 5-10 minutes. So imagine the trees are also facing that. The physical was seen under the direction of the uh, change was also very clearly seen. That means the blast was from one side, the growth rings were shifting to one side. That was also one very interesting observation. So using that, the growth rings can reveal this kind of data as well, right? Yeah.
So, okay, thank you, sir. We have another question from Kanupriya. She has asked, sir, diffuse porous wood can be used for dendrochronological studies.
Yes, it can be done. So diffuse porous means the vessel diameter range is more or less the same, and the growth rings are there, but the growth rings are not very distinct. But if it is observed carefully, you can observe the difference in the growth ring, and some kind of dendrochronological studies can also be carried out, though it is not very distinct as such.
All right. Okay, so Ayush has reframed this question again, and he is asking, sir, my question meant that rings gives season condition. Rings gives season condition faced by a tree in a year. A tree can, if a tree can live more than 100 years, so is that point when its stem stop widening?
No, see, basically when it is a growth ring, as I said, it is also called annual ring. That means one single growth ring is formed in 1 year. It is a number of years. And in 1 year, broadly, there are 2 seasons. So each growth ring will have springwood and summerwood both. So it will continue to form even if there is some change in the seasons. The slight difference in the growth rings can be there. Formation of the growth ring will stop only when the plant will die. I think it's clear now.
Yeah.
Right? Yeah.
I hope this, the doubt of this. But we have one more question. Okay, it is from waiting room. Although, why the stem heart of some plants disintegrate completely and others not?
Stem heart in the sense the central portion of the stem, I suppose.
I also suppose he is asking
Uh-huh.
for the same thing.
Yes. So disintegration of the stems, the cells of the wood, it depends upon the wall thickness is very high, highly lignified ones. So their disintegration takes more time as compared to the wall thickness of some other cells. So overall, what kind of elements are present, what kind of wall thickness is present, the disintegration accordingly varies. And of course, it depends on the conditions also where the disintegration has to take place. If it is in evergreen conditions where there is good amount of moisture, good microbial activity, the disintegration might be faster. So that depends a lot on these conditions.
Right.
Yeah.
And the next question is from Govardhan 209. He's asking, sir, in case of rubber tree, in which position the lenticephorous vessels are situated?
Okay, rubber tree Hevia brasiliensis. So it is present in the sapwood region, along longitudinally present along with the vessels and the fibers. There are these latticephal ducts which are present, and if you take a section and observe, you will find them along with the ray parenchyma also. There are specialized ray cells called latticephers, which are present in the longitudinal portion. That's why when you give a cut from outside, the rubber lattice simply oozes out.
Right. And that is why people used to cut, may make a cut mark over there and collect the
Yes.
rubber from
Correct.
these latticephorous ducts.
Right.
All right. Thank
I
you.
think Gurjant is asking about story or evolution history of plant stellar system. I think this I touched upon yesterday in my lecture in the beginning, when the plants invaded from water to the land during especially the Carboniferous era. So the pteridophytes suddenly gained height, and then there was maximum height gained during the evolution of gymnosperms because of the lignified tissues and because the conduction was being possible. That's how the evolution proceeded. So it is ultimately a result of adaptation followed by competition and all how it all decided. Right. So it's a big story. One can read a lot on it. Yes.
So obviously, it is very interesting to know. Actually, see all the evolutionary history stories about all the organisms and characters. They are very interesting.
Yes.
Because it took them like millions of years. They have adapted those conditions and gradually they have modified themselves and gradually that character becomes that change has become at a genetic level. So that's why it is regularly we can see those characters now as permanent and we can see them in every generation.
Correct. And there, sir, you remember like we had actually met at that workshop at Kolhapur. The theme of the workshop was the evolutionary morphology of land plants.
Evolutionary
entire story of evolution. We got a completely different outlook because of that. Right. So one has to think from that perspective. Everything can be correlated with evolution.
Yes, obviously. And the thing is, everything is see. The thing is, when we discuss about content from evolution point of view, then everybody is right until unless he or she can justify themselves in the light of evolution.
Correct. Evidences. It's all about evidences. And then there can be somebody who can come up with something new which might be contradicting some earlier research. Could be a
Right.
possibility. So there's no question of right or wrong over here. It's just observation and the evidences which are available.
That's right. That's basically right. So this is what we actually, do in when we find something or when we research on evolutionary histories. We
Hmm.
all see we have lots of data available in terms of DNA data as when we
Hmm.
say. So
Right.
everybody is analyzing that data in a different model when we
Hmm.
say the phylogenetic relationships of organisms, and
Yes.
everybody is using the same genetic sequence, and they all are having another different approaches, they all are having different conclusions. That
Correct.
is all just because what they think and how do they think and what evidences they have.
Exactly.
So it's all about your own understanding, it's all about
Right.
how you can relate or these characters or things in light of evolution.
Correct. If you see the genetic level, then sir, you agree that all the entire biological world is ultimately based on these four letters of the DNA sequence and their
Yes.
permutation combination. Everything else is the diversity arising because of all that. It's like nature has been a two great master in
Right.
creating this diversity. Which-
Yes.
This is just one aspect of it.
So
Yeah.
it reminds me all about all those lectures by Dmitry Sokolov. He
Yes,
discussed all
yes.
these points of evolution during
Right.
that workshop, and I think that was the time of 2015 or 15, I guess.
It was 2015. Yeah. Yes.
All right.
September, August 2015. Yeah.
Yes.
Yes. That was
Yeah,
a
so we
wonderful
have some
occasion.
more questions now in the queue.
Sure.
Uh,
Yeah.
we can measure wood decay.
Okay. See, there is nothing as such to measure in the case of wood decay. Some samples can be taken and it can be analyzed. How many living cells are remaining? How much of the decomposition of the cells have taken place? And also, there is no proper asset measure to do it, right? Apparently, or even with the help of physiological analysis, one can find it out. And if it is decay caused due to some bacterial or fungal growth, then one can measure by the amount of microorganisms present in it. That can also be a probable measure then. Yeah.
Yes. She is asking, sir, is there any relationship between wood color and locality factor?
Uh, Color is basically depending upon the type of the deposits which are present in the woody tissues. That can give rise to some characteristic color. Locality as such does not play a role. Indirectly, if you say, could be a possibility that what type of soil condition, what type of minerals, so giving rise to which type of physiological pathway, all that could be a possible connection. But otherwise, it is just based on the type of deposits which are present and a genetic feature then.
All right.
Yeah.
So another question is from Sapna 433. She's asking, sir, which aromatic oil is responsible for strengthening of wood in Tectona grandis?
Okay, so first thing in the case of teak wood, Tectona grandis, the strength of the wood is not because of any aromatic oil as such. As I said yesterday, also the strength basically depends upon the specific gravity or the density and the arrangement of the wood elements. So, how the fibers and the tracheids and vessels are interfused, that gives rise to the strength. That determines the strength. So, there are properties like this extensibility, flexibility, compressibility that determines how the strength can be arising in a wood. The secondary metabolites like aromatic oil and all cannot play a role in.
All right. So they are asking that is the presence of pits enough to prevent embolism mechanism too?
Okay, see, embolism means like if there is some presence of an uh, air bubble or something like that. Pits are the ones which play the major role, and sometimes vesturing and all can be a help in such case. Or sometimes there has been something called as coalescent pits, which are seen. Coalescent pits means like in the intervessel pits that we saw, some adjacent pits can fuse together and form one big opening. That's called coalescence. That has been observed as a mechanism in order to reduce the effect of embolism or something in the case of plants. So this also requires more studies. A lot of work is not done for all this actually. So coalescence can be one probable answer. Again, it is based on facts.
Okay. So now we have the bark is quite thick. What can be the possible reason for this type of thickness of bark?
Okay. Now the bark thickness is again dependent upon the secondary growth, the amount of secondary growth that takes place, the amount of activity of the cork cambium that can lead to the thickness of the bark. And as I mentioned yesterday, that for every species the bark thickness is different. So for wine bottle corks, if you are asking, so we generally select some certain species which are having a thick bark so that this can be taken quercus, right? So the thickness of the bark is basically because of activity of the cork cambium and the extent of the secondary growth. I can see Atul is asking some interesting questions.
Okay, so I'm going to the next question, and I think this is the last one for this session.
Okay.
Atul has asked, does a bonsai technique affects ring and wood formation?
That's a good question again. See, bonsai means miniaturizing the tree species, right? So when you say miniaturizing, right from the growth stage itself, you are training the plant with the help of those wires and all, so that the plant grows only to some extent. So when you give that particular training to the tree species, the growth of the wood and the ring does not get affected internally. Because if you see a bonsai, the plant looks exactly similar to a normal plant, but a smaller version of it. Okay, if it is a fruit bearing tree, it will produce the fruits, the flowers, everything. So the ring and the wood formation is not affected in that. Only thing is that the distance between the growth rings might become less because of the physical intrusion. That's the only effect one can probably observe. So that can also become, in fact, one very good research activity.
That is experiencing
You
all,
can take
all
samples
the,
from bonsai and you can carry out research whether the wood is affected or not. Right? So these are really good ideas that students are coming up with.
So do we have any other questions from the audience? One question again, that is there any machine present to extract fibers?
Say for extracting the fibers as such, there is no machine. The maceration is one method where you can separate it out, or it's like in the case of say jute fibers and all, very economically important ones. So there is a retting technique which is done, okay, immersed in water, soaked in water, and then the fibers can be just separated out. But from the wood sample, just to extract the fiber, maceration is the only technique. There's no machine as such.
Yeah, all those techniques of extraction of those fibers are manual. We have seen all these things in Kerala and Tamil Nadu. People
Yes,
to extract all those fibers from coconut husk as well, and they make some ropes out of them. So they all process all these things manually
yes, right.
in the local areas. All right. Kanupriya is asking, sir, does wood science affect climate?
I think it could be other way around, whether the climate affects wood science, right?
Yes, I don't think science can affect climate. It is all about if there is any. Science is a it's a very
Actually,
broad term
it's a
to use
broad
wood science,
thing.
which is affecting
Yes.
climate. So,
Yes, and as far as the wood structure is concerned, the climate, the change in the weather environment does play a role. But as I said, it is a very highly conservative aspect when it comes to wood. So because of change in the environment, change in the climate, there is not much significant change in the wood. based on the studies that are done till now. Okay, there might be slight shift in the range of the wood structure, but to a large extent, it does not change much. And that's why still we are relying upon that for authentic species identification. Okay, so more work can be done. Like there was this work done by Dr. K.S. Rajput sir on the effect of the wood samples from around fertilizer factories were collected and they were analyzed. So there was some slight changes. But again, it is leading to the same conclusion that the wood structure is a highly conservative feature of the wood. It does not change so easily. I hope this is clear now. You can read more on that. Yeah. I think.
I think I am having lots of connectivity issues here. I can't hear things properly.
Okay. I think we are done with all the possible questions. And if the students require my contact and also they said you can provide it to them, the interaction will go on
I think
endless.
that will be good if you can share the front page of your slide so that they can note down the information.
I'll be providing some possible study material out of these 2 sessions. It will be useful for the students.
Right, that would be great.
Yes, sir. So maybe in a day or 2, if I do that, is that okay, sir? By this weekend?
Yes, that will be okay.
Yes, sir. Thank you.
Because I think the students,
yes,
they are learning a lot of things from your lecture, and all those high quality image photographs of anatomical views of different sections and different plates and tissues of this thing. They must have enhanced their knowledge and their understanding level as well. So I believe they must have enjoyed all the sessions, and now they have a better understanding towards wood and its anatomical structures as well, and basically the not so broadly touched area as a wood science in curriculum these days. So definitely this lecture it must have enhanced their knowledge a lot, and I hope that they have made their notes as well. But in that case, as our speaker, Dr. Vishnu Prasad, he has suggested that he will be providing some notes for the students so that their enthusiasm cannot fade away, their motivation and their knowledge, it remain active, and they can learn and they can explore things in a better way. So he'll be providing you some content. Okay, thank you so much, Dr. Vishnu Prasad, and we will be happy to have you here, and we'll hope we'll meet again.
Hope to meet you all soon. Yes, sir. Yes, sir. Surely we'll meet once again. And thank you very much for providing me this opportunity. It was indeed a great pleasure interacting with students from different parts of the country and sharing this knowledge, sharing my experiences. Right? So it is good, and I wish the best of luck to every student, all of you. Do well, explore well, come out with really interesting findings. And the research, this kind of research and all should go on continuously. It should never end, right? Make a mark for yourself, everyone. All the best, everybody. Blessings to all, and everybody take care, especially in this COVID situation. Be safe, take care, stay healthy.
Thank you, sir, and same to you.
Thanks a lot. Thank you very much.
Thank you so much, Dr. Vishnu.
Thank you, sir. Thanks a lot.
Okay, sir. Take care.
Okay, you too. Take care. Take care, sir. Yeah.
Okay. So, students, meanwhile, as we are done with today's lecture by Dr. Vishnu Prasad, we have another topic which is on digital herbarium. As I discussed with you in our first or second session.
Okay, sir.
So, are you guys here, right? So, as I discussed in the beginning or in the third session, you people were asking about something related to digital herbarium. I read many questions in the chat box that day, but due to the connectivity issues, I could not connect at the time. So today I'm going to give you a brief, hands-on that how you can access the digital herbarium. Okay. So in that case, I'll show you that how we can access the digital herbarium, and I hope you people are aware about certain things like index, herbarium, etc. Okay, is my screen visible?
No, sir.
No, sir.
No, sir. No, sir.
No, not visible.
Now,
No, sir. Your screen is not visible.
Yes, sir. Now it's
Now
Yes, sir.
visible, sir.
Visible.
all right. So
Yes,
you
visible.
can note down this website, that is sweetgum.nybg.org/science/ih. So this is a website maintained and regulated by New York Botanical Garden. It is of index herbarium. Okay, so here at this platform you can access all the herbaria and you can get all the information about the herbaria. Okay, so for example, this is the index herbarium, and I hope you people are aware that index herbarium is a list of herbarium which are spread across the world, and what the information they have, their link, their catalog details, all things are here. If you scroll down into this website, you may come with this platform where you may it will ask you to search the herbarium. So basically this is a herbarium search bar where you can search herbarium by its code. Code stands for the acronym. You all are aware that every herbarium of this world, they have been assigned a particular acronym, and that acronym is a 1, 2, 3, or 4 or 5 letter, or 5 letter abbreviation. Actually, this is a code, or simply known as the acronym. So you can simply write down the code or the acronym here and you can search it. If you don't know the acronym of that herbarium, you can use this for this search bar as well to access the digital or the virtual herbarium. Okay, so you can use all this data. You can put the institution, that if you know the details of that herbarium, that where it is located or in which country it is located, which city, or if you want to search with the name of a person, then also you can write about the detailed search with the person search. Okay, you can write about the first or last name followed by the herbarium code, etc. If you have this information, then you can access all these details, right? For example, uh, let's uh, let's go to meet let's go to the catalog of the Q herbarium. As I entered the K is the acronym of Q, or the Royal Botanic Gardens Q herbarium code, right? So this is the herbarium code K, and it is showing me an option to click into it, right? The institution is Royal Botanic Gardens, while it is located at UK, England, at Q. So in this way you can access the herbarium. For example, let's say I'm clicking on this code and it is taking me to the herbarium details of this herbarium, right? Okay, so now we have the information of Royal Botanic Gardens Q. These are the herbarium details. That Okay, so this is the overview. Okay, so the name of this institution is Royal Botanic Gardens. Its code is K, and current status is this is active. Active stands for this herbarium is working, and it is working, and it is accepting and imparting knowledge. It is accepting the specimens. Okay, so all the facilities offered by this herbarium is active. And the correspondent is Alan Patton, which is the head of collections. So you have the email ID of this fellow as well. So if you need any information regarding the herbarium or any specimen, or if you need any collection, then also you can mail him, or you can email the herbarium at q.org. So there is a method which is commonly known as the loan method. Loan method is a method by which you can get access of the specimen in physical form. That sample can be can be forwarded to you by with the help of post or other services or other courier services by the herbarium as a loan. Only if your institution or where you are working or the country has any understanding with the institution of this thing, the Royal Botanic Garden. So if both the institutions are in this kind of deal where they can share the information as loan, then only you can access those herbarium sheets. Otherwise, you can access that you can access those specimens virtually only, right? So it has all the information, like this is the address and this is the web address. So these days we know the importance of web address and how we can use this information. Okay, so if you click on this website or this address bar, it will take you to another web page, which is actually the web page of the Kew Herbarium. So here you can see they are representing lots of collections. So they are giving a chance to explore the Kew Herbarium virtually. Okay, so you have lots of options here. You can anytime you can go and search about and you can explore these things. It will really give you a lot of content, and that content is actually the quality content. So when it comes to the collection, so they have over 8.5 million items in this herbarium, including all the collections. So it's a huge number. So look at this photograph. They are representing, they are giving you a simple look of their herbarium, that how how you can access and how this place actually look like, right? So they have collections from different, from different versions or different formats or different fields. They have the herbarium, which deals with these pressed and dried specimens of plants. They have a fungarium as well, where you can get lots of fungal specimens. Just like herbarium or xylarium, they have a fungarium, and they also have a very good seed collection, right? So in this way you can access. So here we have the links to explore the herbarium or explore the fungarium or seed collection. Apart from these collections, you can also access the economic botany collections or DNA and tissue bank. So let me tell you one thing, that it is not possible for everyone to get the plant specimen from that region, and sometimes it is also not possible to provide the specimen sheet to everybody for study. And sometimes, as we know that nowadays we are following the APG4 system of classification, and that system of classification is based on DNA data. In that case, if somebody wants any DNA sample from the herbarium, they can also give you the DNA sample which they have extracted, and they'll amplify that region or that gene. They will amplify it, and then they can share that gene or that sequence with you, right? So usually it's 10 vial of 5 microliter to 10 microliter. Okay, so though it is very much costly, but this is another way of of giving information. So DNA data these days is very important, right? So they also have microscope slide collection, where you can see the anatomical structures, as you just saw certain features in the presentation of Dr. Vishnu Prasad. So you can see some beautiful microscopic slide collection as well, and you can explore them here. So this platform provides you a lot of content, right? And apart from this, now you can also explore the spirit collection. Okay, the collection of the specimens in 70% alcohol or other in the jars, or the very big specimens which are there. You can also explore those collections. You can also explore the in vitro collections and the library as well. And let me tell you, the library section of this herbarium is very, very interesting, because it will not only give you the literature, but it will also give you the letters which actually passed between scientists or people at the time, like J.D. Hooker or Bentham and all other people. So at the time when there was a time when Britain was ruling almost around 80 countries. So, and they were exploring the flora and fauna of all those 80 countries. And in that case, the email or services or the postal services, it was not so strong, and actually the technology was not there. So at the time they used to write letters to their seniors or to the experts in England or anywhere else. So in that case, they used to draw, they used to make illustrations of those plants, and they shared the letters. So you can get the original copy of those letters. You can see that how they used to communicate, how beautiful they used to paint, they used to make those illustrations. And many of you must have seen these illustrations in Forest Research Institute, because there are, so that institute or the CNH, they have lots of collection of these kind of illustrations or letters as well. So they see, this is the illustration and artifact collection. They have archive collection, which where you can get all those letters which I'm talking about. Okay, and these type of illustrations they used to make it at that time when there was no camera or something was available. So they used to draw that entire plant, and they used to send that paint or drawing to the expert, whether it is in England or in Australia or somewhere else. And that postal services was also run by the ship postal department of Britain. So we are going to the herbarium part only, and I will leave you with this website and the mechanisms where you can explore all possible collections, whatever you would like to. But I'm going to look into the herbarium section of this thing. So if I'll click on explore our herbarium, they will take me to the herbarium section, which has around 7 million preserved vascular plants. Okay, so it is indeed a very huge collection, and they have collected it in past 170 years. As I told you, since when they were actually exploring the different parts of the world, and before them actually Linnaeus and his students, they were exploring different parts. Because as I don't know whether you people are aware of this thing or not, but many of you must be aware that Linnaeus, he never traveled. His students actually traveled in different continents and different countries, and they used to send him the specimens, and on the basis of those specimens he wrote the species or genera Plantarum, right? So this is the Kew's herbarium. You can see the video over here. If you click on this, this video has been also put it on YouTube, so you can explore the herbarium here. Okay, and so this is, see how multi-story platforms or levels are there, where all these cabinets are kept, and in every cabinet there are lots of specimens are they are present there. So they have preserved a lot of specimens, and this area, this central area, is basically used to study those specimens, right? You can take out the sample from these drawer or basically these pigeonholes, and you can put them here on these tables and you can study them, right? So these are the platforms to study the specimens. All right, so you can read the history of herbarium, and it is also in your syllabus of second year, I believe, when you learn the techniques or basically the herbarium sections. So these are the herbarium specimens. Okay, so let's see how we can explore the herbarium specimens. You can get all these details from here, what they have written over here, that how they collect the specimens, and these days how they are using the DNA samples, and how they are extracting DNA samples from old materials as well, right? So you can explore all these things. So now here at the bottom of this page, we have the link to access the herbarium, right? So this is the link. If I click on this link to access the herbarium, it will take me to the catalog of herbarium. Okay, so from here, either you can book a research visit in a physical form to the Royal Botanic Gardens Kew, or you can access the herbarium online. Okay, and also you can find how to arrange loans, as I just discussed with you about the loan method, but it can only be has some understanding with the Kew, right? Okay, so here I have the link to access the herbarium catalog, from where I can actually look into the specimens. So this is the herbarium catalog of Royal Botanic Gardens Kew. Okay, from here you can search in this search bar either with the scientific name or family or country, or you can also go with the advanced search tool. In advanced search, we know about the details of the specimens. If we know all the details like the family or the genus or the collector's name or the country or the barcode. Even if you have any information, you can simply search for these plants. Okay. All right. So for example, if let's say we are looking for the family Fabaceae and a genus. What genus we should look for? For example, let's take Delaunus, okay, and Regia. Let's see if we can get anything. So we have retrieved 5 records, belongs to 1 family in 3 countries, 1 genera, and there is no type specimens, but we have lots of, okay, we have this much of specimens, okay, from different countries. So you can see the location. They are from Mexico, USA, Brazil, and then USA, collected by different people. Okay, and this is okay. So this is the spirit collection type. It is not a herbarium. It is not in the herbarium format. Okay, so in that case, we can look for the barcode ID as well. See, these are the details of the specimen. Okay, and the determination history as well. So you can look at all this information from this platform. Okay. Okay, there is no image available, but it's okay. So we are going to look at a simple search. For example, I'm searching for Solanum nigrum. Okay, and if I click on go, then I have some results. They are showing that they have retrieved 794 records from 1 family, and the collection is from 104 countries, 1 genera, and 4 type specimens. I hope many of you have studied about the type concept and are aware about the types. So we have this much of samples, and we have around 10 pages of these samples. So I'm giving you an example by okay, so just look at this specimen. So here we have all the details of this specimen, that this specimen belongs to the family Solanaceae, and its current name is Solanum nigrum, subspecies nigrum. The collector is first P, collector number is 2821, and the collection date is 25/6, but they are not sure about the year because it has been written only 62, so it could be 1962 or it could be 1862, and it was collected from Iran, and the location is Kalas Gorge. So let's look at the specimen. So when you click over the specimen, they will take you to this. Okay, so now look at this sheet. There are lots of content which we have to look and we have to understand for. So every specimen of the Royal Botanic Garden or any other garden, they are having their barcode these days. So now we have technology. Now we can access all this information through a digital format, because when a herbarium is having more than around 7 million specimens, then it will be very difficult for them to find out a single specimen from those bulk of specimens. In that case, they have this barcode, and by entering the barcode or by entering the name, or even if you have any, if you want to look at any particular specimen, for example, you are looking these specimens online, but you would like to see those specimens in physical form, then you will go to the herbarium and you will ask for this barcode, that I need a sample with this barcode. Then they will enter that detail in their in their database, and they'll find out that where this specimen is actually placed, so that in this way the collection in this way the easy for them. So you can see this specimen, and there is a label that it is from flora of Iran, Solanum nigrum, and see all the information they have written over here, that what was the this of width and 2 inches is stem leaf itself about some. Okay, so they have some measurements. They have details of the flower, or basically we call them as field notes, along with the collection number, that is 2821, collected by P first, and on 25th of June 19 or 1862, because they are also not sure that which 62 they are referring to. So today is also 25th June. What a coincidence that today we have this specimen with us. It was collected on 25th June of 62, and that 62, I believe this 62 will be of 1962 maybe, but let's see. We don't know about exact day, exact year of this 62. And at the bottom of this you can see there is a scale, and along with the scale you have some color codes, right? So these color codes are actually they are universal color codes, and on the basis of these color codes you can identify the color of the leaf or stem or any other part. So because we all can see, or we all can identify colors in different wavelengths, right? But there are certain color codes for the universal identification of colors. So these are the color codes, and this is the scale which you can use to measure the leaf size or the other or for to measure any other part of this plant, right? So in this way you can access the specimens. Okay, if you click on the full size image, you can so again you can simply zoom it out and you can study the characters. Okay, so these barcodes and other information, they are very important sometimes. I hope you are getting at how to access the specimens from Kew Herbarium. Yes, is it making any sense, guys?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, so you have lots of information here as well. You can look for the different projects which are going on in this herbarium. So Kew is also, this Kew or the Royal Botanic Gardens Kew, they also have some programs in which they train the students, or they also offer certain courses like MSc and PhD as well. So one can also, if they wish, they can opt for these courses as well, and they have lots of content. They have lots of programs, and they keeps on. So they every year they organize lots of programs about biodiversity, about conservation, plant identification, and not only plant but also for fungus and other things as well. So okay, this is the thing. For example, if you would like to see any other plant, you can just put their name in this search bar and you can access the material. Okay, so for example, what should I? Okay, for example, I'm searching one specimen, Indigofera dalzeli. Okay, so now we have one specimen here. Okay, so we have retrieved 2 records from one family and one country. Why one country? Because, and see, this specimen which I just asked for, I just requested from this website, this is endemic to India. And when this specimen is endemic to India, so its collection, its region of collection is only one, that is India, right? So now you can see this is the scale bar, and can you see the copyright reserved mark here? That means if you are using these specimens or these virtually accessed specimen and you are using them for any reference or for anything, and if you are quoting these specimens somewhere, or you are using these photographs, then you have to take permission from the Kew Botanical Garden, right? Because all the content is reserved under Copyright Act of IPR. Okay, so now you can see that this specimen was collected by N.A. Dalzell, and I think you can see there's something, it has been written. This is the Indigofera triquetra dalzell. Triquetra was this, the earlier name of the earliest name of this plant when it was discovered by Dalzell. Okay, so this is the information. These are the color codes, and see the specimen. So how they used to have, they used to preserve these specimens. And if we look into this label, can you read? So this was presented by the wife of N.A. Dalzell, Mrs. Dalzell, to the Bombay Herbarium in April 1878. So it was donated in 1878, and this plant was actually discovered for the first time in 1860s. Okay, from Maharashtra. So now even today this plant is this plant, its growth and its presence is restricted only to the Western Ghats of Maharashtra, Goa, and Karnataka only. So, and this plant is endemic to India. You cannot find this specimen anywhere else in the world except the Western Ghats. Okay, so we have some lots of example and lots of specimens which was collected. They were, so if these specimens were collected like in 70s or in 80s, so they have their dates. And always remember this thing, that we always write all this the information with the help of pencils. Why? Because this pencil cannot be removed by alcohol or any other kind of solvents or anything. So sometimes we also get these specimens with the same handwriting of the collector. Sometimes we access these specimens of Linnaeus. Sometimes we access specimens of Roxburgh, of Gamble, or many other eminent collectors who have collected specimens from different regions of India as well. And there is another platform which is called JSTOR. You can actually look at those specimens in JSTOR, and you can actually measure the length, width, and other you can also measure the dimensions of the plants in that platform of JSTOR, right? So this is all about the digital herbarium, I would like to say. And if you have any query relating to digital herbarium or virtual herbarium, you can always ask me. Okay, just feel free. So I'm ending this session here, I hope. Okay, so there are certain questions again. Nisa has asked, how can they keep the DNA viable for such a long time? See, when it comes to DNA, we preserve DNA in two different forms. If you want to work on that DNA in a near or in the near future, then obviously you can keep that DNA at 4 degrees centigrade. But if you would like to keep that DNA for a very long period, you want to keep it, then you have to keep it in minus 80 degrees Celsius, right? So DNA is usually, if you are not using, or if you are not fluctuating the temperature frequently, then the DNA will remain there. And it is nothing about viability and something. See, we always say that if the DNA sample or the gene which you have just isolated or you have just amplified, if you want to give that gene entirely, then you have to take some care that you should not, what do you call it? You should not fluctuate the temperature where you are keeping it, because it will shear the DNA. Second thing is, you don't have to rigorously shake the vial of the DNA. So you have to keep it gentle, you have to keep it at low temperature for long-term storage, and obviously it remains like that only. And when Q or other organization, when they share, or when they pass, or when they parcel these kind of samples of DNA, they put them in special baggages. There are many coolers or many ice coolers are also there, where we can maintain the minus 20 Celsius degree temperature, and we can simply pass on these things. So
Thank you, sir, for the answer.
okay, so people are asking that what is the time period for the time duration of the test. So let me tell you these things very clearly, that we will have, I told you that we will have 30 questions, but now I am revising this thing, that we will have 25 questions for every test, and that test will include all the content delivered by us. Okay, and secondly, that test will be of MCQ type. It will be of maximum 45 minutes, right? So you have to submit your answers in maximum 45 minutes. Is that okay to everyone now?
Sir. Excuse me, sir.
Yes.
Sir, will the test be in Google Forms?
Yes, it will be an objective objective one, and it will be most probably in the Google form format.
Okay, sir. Thank you, sir.
Okay.
So are you going to share the link of the test with us?
We are going to share the link of the test with you all, at a common platform so that you all can get the
Thank you, sir.
Okay, thank you, sir. Thank you, sir.
Okay, take care all of you.
Thank you, sir.